AI discovery, AI and drug discovery. And it will be a very controversial presentation, so be nice to me and I will try to be as nice as possible to you as well. Before I introduce you to the workflow of the whole presentation or the points, I would like to set a framework. I would like to actually communicate the basic idea about AI in drug discovery to you, which is also kind of like a reflection of the status quo. So I've collected a lot of examples that I will go through to give you an idea of how impactful it can be, where the advantages are, and where also maybe pitfalls may appear here and then. Everything I present to you is a very subjective perspective from my side because I'm a medicinal chemist, and the judging is completely up to you. So sit down, lay back, and enjoy the whole session. The first topic we will tackle is actually what is machine learning and what is AI. Next, we will go through how to collect data for your approaches, and also the different methods in machine learning and artificial intelligence. The next thing is actually the drug discovery done by machines. We will talk a little bit about the hype around it and the cool reality. And we will finish with drug discovery stories I have collected. Also address the, I would say, problem of novelty in machine learning for drug discovery, and also how the industry adapts. Again, this is only a snapshot, also considering the publications. It's close to impossible to cover this whole area and the whole field with a few slides in one single presentation, given the fact that it's only supposed to last for maximum 1 hour. So take it with a grain of salt, as I would say. And yeah, the first thing I would like to address that AI and machine learning is without a doubt still a part of the computer-aided drug discovery field. So anything where you use machines, models, calculations to predict if a compound is of interest to you is a computer-aided drug discovery project. You can do a lot of things with that. I don't need to address every single point on this slide, but you have to keep in mind that machine learning is only a prediction, and it can be good, it can be bad, but it's only a sole prediction. So since humanity was punished a few eras ago with the collapse of the Tower of Babel, we are now in an unfortunate situation where everybody speaks a different language, right? So there's English, there's German, there's, I don't know, French. And given the fact that even if we speak the same language, we sometimes mean different things while using the same or different terms. So AI and machine learning are usually used in the same manner. So if people talk about, AI will revolutionize drug discovery, in ultimate theory, they also always, or sometimes also referring to machine learning. So artificial intelligence is actually a broader concept where by the end of the day, the whole apparatus, so the whole engine, can come up with solutions on its own. Whereas machine learning is a subset of AI, so it can be kind of like an entry to the artificial intelligence. And machine learning applies different algorithms and statistical models to predict something, right? So we take data, we feed data to algorithms, and then we generate a model that is able to predict something. So again, even during this whole presentation, I will be showing to you now, if I use machine learning, I'm kind of like also referring to artificial intelligence and vice versa. So I think we all are now in a very exciting time zone, time age, where we are getting in contact with AI. It can be applied on different areas. So I mean, who hasn't used ChatGPT to rephrase a sentence for his thesis or come up with synonyms? But it's also penetrating other fields like navigation with Google Maps, also your social media. Social media feed is based on algorithms where things try to learn what to show you, so you stay longer in the loop of the feeds to advertise things to you. Advertisements are getting more personalized. They can even predict what is your favorite color and show you the exact dress. And also even Netflix recommendations based on what you've been watching yesterday in the evening. So AI is now very prominent, and it's maybe also a leading cause why it's also so prominent or becomes even more prominent in drug discovery. But the whole concept about AI is not so novel, at least for drug discovery. The first reports were in the 1950s, were basically the flavor of AI, so kind of like easing up the life, convenience increase in drug discovery has been pursued. And in 1965, the University of Stanford applied the Dendral project, where they tried to construct an algorithm to read organic spectra, or the mass spectra of organic molecules, to predict their structure. With the modern advances in machine learning and neural networks and computing, we can now see that this whole topic resurges in the 21st century, in the past, I would say, 2 decades, where it becomes even more prominent. I think the increase in computing power is a major role, because now every small group even has a small server farm standing somewhere in the corner that can process a lot of data, can be used to also store a lot of data. And also, yeah, the convenience of having access to a high performance computing via Amazon Cloud and so on is also something that kind of like, yeah, nourished the whole idea of implementing AI in drug discovery, because it works with a lot of data and requires a lot of computational resources. So, how does AI work? If you would completely boil it down to a very, very, very simple idea, it would look like this. The first thing you have is data. Then you transform this data. You use your transformed data to train a model. You check your model if it does what it's supposed to do, and then you make decisions based on the outcomes of the model. If we boil it down a little bit, it becomes a little bit more complicated. I really like this term, data is the new oil. And I think it's kind of like really reflecting how things have been evolving in the past decade based on social media, where every single word you use on social media can be used to predict your behavior, your consume behavior. So this is also where kind of like the whole thing comes together. Because the more data you have, the more you could actually extrapolate. But you don't need to process everything. So when you have the data, you need to collect this data, and then you need to prepare the data. You need to select, what do I want to focus on? Which parts of the data are of interest to me? Do I would like to work only with active molecules? Then you need to do some sort of filtering. Once you have your data in place, you need to transform your data. What does it mean? Not everything that is data can be read. Right? So let's assume you would have a picture of, I know, something cute, like a dog. So you have a dog picture, and you would like to use the picture for machine learning. A picture is something that cannot be easily processed, right? So you need to transform it into something that can be read by programs, algorithms. So given now the structure of silicon chip at the bottom, we have different possibilities to represent the structure. So you could provide 2D data of that, but then you would actually do the same thing. You would try to check for the 2D and process this image into something like readable, like smiles or fingerprints. So fingerprints can be read because it's just a sequence of zeros and ones. Smiles, on the other hand, is also a readable format because it's just a sequence of numbers, of symbols, and also of letters. Once you have your data transformed, you use the data to train your model. So you choose a model or an algorithm you would like to apply on your data set. An important part is here to also split your data. So basically, data you would like to use to train your model, and then data that you are aware of the properties to use to check for the quality of the model subsequently once your model is finished. Then your model runs whatever it's supposed to do, it learns patterns, and then finally a model is created. Once the model is created, you obviously need to assess and evaluate the prediction quality of the model. So you confront the model with unseen data, which can be the testing set, and also novel data. And then you assess the performance. Can my model predict actives from inactives or not? Finally, you need to make decisions. So you deploy the model for usage, you feed it with new data, and then actually use it. And this is where some companies struggle. because, yeah, sure, you can construct a model, but you also need to get access to this model. So you actually need to use this model in an environment where it can perform. It doesn't make sense to just construct a model and only you can use it, but if you can also include this model into an actual workflow where everybody has access to this model, it greatly improves the performance of the model and also the outcomes, and finally, it makes your life better. Finally, you should also monitor and update your model. Every time you get the chance to actually feed it with new and improved data, use it because it can and will improve the quality of the model. The last four entries here are actually part of an iteration cycle. So if you are not satisfied with the performance of your model, it can always make sense to adapt your model. So you do some hyperparameter tuning, which means you adjust the weightings of connecting points, you adjust the data here and there. You can also feed new data if you say, well, I fed only 100 actives, maybe I should use more to also improve the model here and there. Sometimes it can also make sense to see, well, my model is completely underperforming, the results are more or less random, where you can also decide to go for another algorithm to check if this one would be more applicable to your use case. Data sources. So data sources are actually the bread and butter of data scientists and machine learners. I don't think I have to go through every single one of them because they also address different needs. The most prominent one would be the Campbell database, which includes data on bioactive molecules, which is kind of nice because if you would like to develop a model for a target that is now of interest to you, you can go and check, is there anything published already, and then just mine for anything that is active and also very, very important, inactive, to feed your model with the data. So this is one list. You can also take a screenshot or just wait for the YouTube upload. And there are also more databases obviously available that can be of use in regards to whatever you would like to achieve with your machine learning approach. So next I would like to talk a little bit about the machine learning methods. In ultimate theory, you could subdivide them into two parts that can be also combined a little bit. The first one is supervised learning. Supervised learning is kind of like using data that is labeled. This is the most important identifier for supervised learning. Labeled data means that you add a value on characterization of an entry. So you have molecule A and you say molecule A is active, molecule B inactive, molecule C active. You can also provide numerical values like IC50 values, EC50 values, Ki values, you name them. It can be a lot of things. What you can achieve with supervised learning is regression. So I think we can all recall things like the prediction of compound activity. Saying on the x-axis, you could, for x1, you could have a principal component or even log p value. And on the left side, you could have another value like activity. So you could actually check for how does the activity of my compounds correlate between log p value and activity. And then use this to extrapolate for prediction of novel compounds and their activity. I think we all have been in the situation where we've been challenged with QSAR, so quantitative structure activity relationship analysis. This is one of those kind of things where you just predict the activity of a structure based on prediction. You could also use it to predict log p values, and machine learning algorithms have reached a status where their log p predictions actually even better than the experimentally measured values. So this is where machine learning has actually reached a very, very satisfying status. On the other hand, you could also use for classification, which is the right figure. You could differentiate between active and inactive molecules, kind of like building clusters of compounds. You could also use this to predict toxic or non-toxic compounds. On the other hand, we have the unsupervised learning. Here, the input data is unlabeled. So basically, we don't actually know a lot about the data. Yet, we can use it to kind of like cluster compounds. One usage for that could be biological activity. So let's assume you would have a set of compounds with different parameters. And here we could say, well, the blue ones are dopamine receptor antagonists, the red ones are dopamine receptor agonists, and green ones are histamine receptor agonists based on the input data we provided it with. And if we would then feed the whole system, the whole model with a new compound, we could actually deduct if this belongs to one of those three cases. Another idea would be to use clustering for molecular scaffolds. So basically, not only looking at the 2D structure, not only the kind of like obvious fact, but also to look maybe a little bit into the topological side, like the volume and orientation in the room. Another nice thing is actually the association. So that you could associate a particular motif in a molecule to a biological activity. So can you correlate the presence of a pharmacophore to a desired effect of your compound? Furthermore, it's also something interesting that can be used for the prediction of synthesis route and adverse drug reactions. So you see, maybe just also a little bit of background. I'm a little bit focused on the medchem perspective and small molecule design. Obviously, there are a lot of things that would be obviously in the higher field of drug discovery, kind of like phase 1 and phase 2 assessment of biological data, but this is not what this whole presentation is focused on. Now this is where the fun part starts, and this is also where a lot of people drop the ball. When we dive into the deep learning. So deep learning is a little bit more abstract. The key feature of deep learning is that you enter a high dimensional data field. So let's maybe start with a top example, convolutional neural networks. So convolutional meaning stemming from evolution, and so evolving together. You take a picture. Yeah, so convolutional neural networks work extremely well for pictures. You take a picture, and then you go through the picture and you tell, well, what's the presence here? What's the color? What's the shape? What's the depth of this particular area? And then for each section and each area of the image, you construct one of those little representative numerical values. Those convolutional layers are then stapled. And then what the idea behind this algorithm is, well, are they all really necessary? So you actually reduce the layers here to a smaller number. So you generate a pool of features that still kind of like capture the whole idea of the image, but are smaller, include less dimensions. And then you keep on continuing. You look again at this pool, and then reduce it to another convolutional layer, and then check, well, what can we remove? And so on and so forth. And you do it a lot of steps, a lot of times. Finally, you end up with a final pool. And what you do then, you flatten the pool. So basically, you try to reduce this multi-dimensional layer into a one-dimensional string. So only one dimension, meaning you will only include only numbers, a sequence of numbers, long numbers, but a sequence of numbers nevertheless. Finally, you go into this fully connected layer that can actually read this flattened layer. So this layer is based on information, the data that you use to train your model. And what it does then, it reads this flattened layer. It then compares what are the patterns of this flattened layer that I can actually read and understand. What happens then? You receive an output. So it will tell you, this is a picture of a cute dog. I mean, like, maybe some of you have seen it. You can train actually one of those CNNs to recognize, to differentiate between chihuahuas and a blueberry muffin. Yeah, so that's kind of like the most prominent use case I've seen so far, at least. Another interesting application is actually the deep learning neural network. Again, you start with an input layer. And what you do then, you construct a neural network. So everything is connected with each other. What happens here, you extract different informations, different parameters from each input layer, and then feed one of those neurons, basically, one of those hidden layers. And then they feed each other again and again, where you transmit one information from one neuron to another. So what happens here again, we have a lot of layers, we have a lot of dimensions going on, But by the end of the day, we again reduce them to a simple representation. And this representation understands a lot of connections happening in between. So ChatGPT is actually a deep learning neural network that uses this kind of approach. So what this thing can do and what this whole deep learning approach can achieve, it can understand a lot of different connections and a lot of different references between different data points. So ultimately, it can connect apple to the taste of sweet without having both of them standing next to each other in a long, long text. Just mining from a lot of different sources, a lot of different input. And what this can achieve is actually it can also detect a lot of things that you didn't explicitly request for. We'll see that a lot in our subsequent examples. So now we have a basic idea about machine learning in itself, a lot of data, a lot of processing. Now let's go back to pharma. So the thing with pharma is we are extremely, extremely bad. No other industry accepts such high failure rates as we do. So basically, drug discovery has an abysmal failure rate of approximately 90%. And this is based purely on phase 1 clinical trials. As you can also see that 90–70% of cancer drugs actually fail during those clinical trials because they are not able to meet endpoints. And there are a lot of things that contribute to that. One of those is obviously the lack of clinical efficacy, basically because the drugs do whatever they do, so they inhibit the enzyme, but once you apply them on a broader scope with a lot more layers, a lot more complexity, because the human body is quite complex, you will end up with a lot of failures. Surprisingly, toxicity becomes also a problem. I think this is relating to the phase 1 clinical trials per se, because people do not expect side effects because off-targets were not realized back then, or the dosage is not as was extrapolated from the models. Surprisingly, which is a good thing, the poor drug-like properties are only amounting to 10–15%, based on also a lot of things that happened in the preclinical trials and the preclinical optimization. What also surprised me a little bit is actually the lack of commercial needs and the strategic planning, but this is not something we as scientists in the drug discovery field can influence too much. What I found pretty interesting is actually that the whole research and development procedure has become less efficient in the past decades. And there are a lot of factors to that. So basically, for every $1 you invested back in 2010, you returned 10 cents on your research and development efforts. In 2020, though, it was already reduced to only 2 cents. And people will ask themselves, well, how can this be? And one major contributing factor is to that the reviewers here, or the scientists, the authors, assumed that everything that can easily be researched has been researched already. Because if you think about that, we have all of those huge high throughput screening libraries and so on. And most of the easy compounds that are easy to synthesize have been also tested on the easy targets. So now, if you still would like to market a drug or get an approval for a drug, you need to put more effort than you had to do in the previous decades. So ultimately, we see that if we could actually improve the prediction to the clinical efficiency, toxicity, and so on and so forth, we could actually save a lot of money and also accelerate the development of future drug candidates. In this little figure and graph, you can actually see how much time we can save. This is an assessment. And on the left side, we actually see the millions that you could save. So here, 500 millions that you could save. And here are the colors. So basically, in the early stages that are more or less here, we can actually see that the speed is not the problem, so we won't save too much money there, but we actually need to improve the quality of the predictions, and this is especially true for preclinical and becomes even more important in clinical phase 2. And actually, if we are able to reduce the costs, and costs could be actually the acquisition of compounds, including the purchase and also the synthesis of compounds, as well as the management. So it would be great if we don't need to synthesize 500 compounds to completely understand the structure-activity relationships, but could only maybe limit it to 150 or even less. And what you can see here, the highest peak is actually the clinical phase 2, where quality becomes a problem. And if we could reduce the failure rate here by 20%, we would actually save up close to $450 million per drug approval. And this is actually where I would assume most companies are now aware of, because this is where the target selection is actually the problem. So a lot of pharmaceutical companies have now restructured how they do the target validation steps here, and they now put a little more of effort to select which targets should be pursued for a particular treatment. So this is actually already an implementation in the pharmaceutical companies around the globe. So now to the AI in drug discovery. So the first coming out of the drug discovery demo. And I think in the past few years, we all have been on LinkedIn or other social media platforms and whatever it may be. And you can actually see how people are celebrating AI to solve every single problem that is out there. I mean, even if the financial newspapers and journals jump up on that, you can actually feel that there is a lot of money flowing in this direction. And it's actually true. So by 2021, approximately 16 billion have been spent on AI and drug discovery relating to collaborations or acquisitions of companies. And those are only the reported deals. So the true number will be much, much, much higher. But if you think about that one FDA-approved drug that costs approximately 2.8 billion US dollars, you could actually ask yourself, well, we have invested 16 billion by 2021 and even more in 2023. So where are my FDA-approved drugs that have been discovered by AI approaches? And yeah, it's a thing, and we will talk about it in a minute. But what became pretty obvious that a lot of activity in the early stages of drug discovery pipeline is actually targeted towards really explored targets. So GPCRs, kinases. And this is no surprise because data is a limiting factor. And we will jump on that also in a minute. But before we do that, I would like to talk a little bit about the hype around AI. So there are actually 3 reasons why AI in drug discovery is now as prominent, has become so prominent. Obviously, the only thing that rules the world is the financial consideration. So since AI companies are now stemming around the world and you see another company popping up every 2 minutes, at least that's what it feels like, there's a lot of money flowing in startups and also in the acquisition. So a lot of companies actually work together with smaller companies or middle-sized companies, middle-sized AI companies to develop drugs and get drug candidates from them. Since I showed you this extrapolation, there is actually huge potential to save money. And it's actually far easier to save money than to make money. So if you could kind of like reduce the costs here and there by 20, 30%, you would still save close to $1 billion US dollars per FDA approval. So a lot of money that can be saved there. Furthermore, the market opportunities. So if AI is able to target novel drugs, novel targets, novel target classes, you would also have access to novel therapies that are obviously of high interest financially. The other aspect is obviously the technological advancements. In the past decade, we are actually now in a very exciting phase where we actually start to understand what AI and machine learning can achieve nowadays with the data they are provided with. So people catch up on that. 5 years ago, nobody was aware what machine learning was. But if you now ask somebody during a conference, what do you think about machine learning? They will have an idea about that, or at least an opinion. The workflows are also a little bit easier to use, so it's even possible for early stages PhD students to design their own workflows. it's more convenient. It's not only like a geek thing nowadays, so it's not completely limited to experts, and even smaller approaches can be done easily from home at your from your standard hardware. Also, automation efficiency is kind of like coupled to the workflows and easier usage. And this is what my personal gut feeling is, that drug discovery becomes more and more interdisciplinary. So different departments work way more together than they used to work together 5 or 10 years ago. The team constellations are different than they used to be. And this brings into a situation where data scientists talk to medicinal chemists, talk to bench chemists, talk to biologists, talk to pharmacists, to whoever. So a lot of people are starting to understand each other. And this is also opening a lot of possibilities, but it's also putting a lot of challenges towards us. And obviously, data processing and analysis has become way easier because it's easier to access a lot of data, to process a lot of data. I mean, like even the Campbell database, if you check what it is now or today compared to what it was 20 or 30 years ago, it's not comparable, and it's far easier also to operate. And finally, the most important thing for us humans, because this is what we love to understand, is the psychology. So people are very optimistic because let's look at ChatGPT. It made a lot of lives so much easier and way more convenient. Why shouldn't be this case for the AI and drug discovery field as well? So we are optimistic because we expect a lot of things to happen in the next 5 to 10 years. Another idea is obviously the fear of missing out. So if a company does not have an AI or machine learning department nowadays, it feels kind of like off because they are not jumping on another hype train and not pursuing this method, this approach. So this is something where people and managers can also say, well, yeah, I don't want to be the bad guy who made this decision. So let's at least try it a little bit. Something very interesting is a positive feedback loop. So it's pretty rare to see a model advertised to be bad and to show people, well, we did a lot of effort, we invested a lot of time and computational efforts and resources, and yeah, we cannot do anything with this model because it's bad. Nobody reports that. Only the good stuff is reported. We can now predict cancer cell lines to be sensitive towards this and that. We can read X-ray images much better, and so on and so forth. So the positive feedback loop pushes the idea of that AI can do a lot of things. And last but not least, since a lot of money has been flowing in the past years, everybody needs a winner. So we need actually to show success. And if the success is not there, then we haven't invested enough money. So you don't want to drop this child and just keep on going until something comes out of that. In this regard, we should all be aware of the Gartner hype cycle, which is kind of like an assessment of every time an innovation and technological innovation comes around, where it becomes actually useful for us. So in the past decades, so since I said machine learning and AI is a huge field, there are a lot of things going on in this area, and also the different positions like, de novo design of drugs, or ADME predictions, or clinical phase 1 outcomes. They are all on different stages on this hype cycle. So once this technology kicks in, it becomes obviously more visible, and obviously the expectation rises up to a point where people realize, oh, we are not there yet, and a lot of people drop out, and only a few companies still continue to actually develop and work in this area. And at some point, the things start to actually be useful. So this is a slope of enlightenment where things start to be implemented inside workflows to be used on a daily basis. And once everything has come to a conclusion, we reach a plateau of productivity where we can actually achieve something and tell success stories with the technologies we've developed so far. So, but before we dive into that, we also have a problem with AI. So AI machine learning needs data. And the quality of the data is actually what dictates the outcomes of your model and the power of your model. So the question is, do you believe everything that is on Campbell? This is something you have to answer for yourself. This is not something I have to answer for you. But we can agree that even doing our PhDs or doing our work with other people, even like biological data, it feels like sometimes some people perform the experiments better than others, and some data was more trustful than others. So quality is a thing, and it's very, very difficult to get access to good qualities here and there. The other thing is, it's pretty hard to assess the power of AI because companies working in this field keep almost everything pretty tight to themselves. They don't publish that much. They show, yeah, we can do this, we can do that. But the cool stuff is actually kept behind closed doors. It's close to impossible to actually get a hold of a 2D structure of a molecule that is now in clinical trials that was actually designed by AI. And the same goes also for the results and further details. This leads to actually the fact that machine learning, personal subjective assessment, gets a little bit overestimated. It can do a lot of things, but we are not yet there where we say that it can do everything, by far not. So what we need, we still need a lot more of success stories to actually trust machine learning in the future better. As I mentioned before, because it's an interdisciplinary field, it's actually difficult to bring a lot of people with different backgrounds together. So data for chemists has a different value compared to biologists, because chemists don't care that much about, the things that biologists would consider to be important. And therefore, picking the right data from a diverse set of people involved in that is not pretty easy, because also you don't know which data is actually of importance to you. Furthermore, we are still far from the point where we completely understand machine learning, which means that if we can process data, it's still not the same thing as understand the data, and absolutely not the same as interpreting the data. So we need still a lot of things also going in those two environments here. From an academic perspective, I would also say that I find it highly problematic to publish things on machine learning, because nobody would go the extra mile to actually reproduce machine learning approaches, which can have many causes. One of them will be because if it's a pretty good model trained on a lot of data, it's close to impossible to be done financially, because you need so many computational resources to do this and do that. Since not everything is published and a lot of things are kept hidden, intransparency becomes a problem here, and also the intransferability of the results for different groups. Because if you use measured log p-values or measured activity data in one group, you cannot compare or use the same model in another group, because they will have different data and also different biological data there. So it's not so easy to communicate different models between different groups. And obviously, the first thing you have to do to actually save money, you have to invest money, and not everybody has the money to do so. This is just a collection of AI methods or fields where AI methods are used. I think it does make sense to address one of them, just to go through the main topics is actually to identify novel targets and actually processing targets or macromolecules. Also, gene expression and genomics is a huge and very important field here. I think we all are more or less in the same area of small molecule discovery, so this is the important part for us, where we can actually use machine learning for de novo design of compounds, visual screening, accelerating visual screening as well, the prediction of interactions, toxicities, even kind of like supplementing MD simulations to run them faster, and most importantly, the optimization of compounds. Also for synthesis, for the bench chemists, chemical synthesis is a field where machine learning is actually doing lots of work, because we can now have very reliable retosynthesis pathways that can be easily accessed, also predict reaction yields and the success rate of a synthesis. Cool stuff that can also pretty easily improve our quality of life there. Last but not least, fields of the post-preclinical phase. So preclinical and clinical development, where you work a lot with patient data, a lot of metadata, multifactorial data. And subsequently, obviously, the safety concerns once the drug has been approved to also monitor adverse drug reactions and so on and so forth. Okay, now let's go a little bit more into the real subject, namely the application. So I mean, everybody has been in contact with AlphaFold. I think it was celebrated. Everybody loved AlphaFold. It was the huge thing. It was revolutionizing everything. It was it was huger than bread, right? It was so amazing and everybody was celebrating. And if you think about that, it's actually pretty cool. So this was the workflow. We don't need to go a lot into detail. Basically, what you need to understand, what was used is actually a sequence alignment of protein structures and then alignment of available 3D templates, and then using all of this to predict novel structures of proteins that have not been published so far. What is beautiful about AlphaFold is actually the attention mechanism that was implemented there, that can actually detect long distance interactions given only the sequence of a protein structure. So it was very powerful. It required so much data, so much processing. It's really unholy if you think about that. But what we ended up with is, I think, the most reliable source for really, really, really good models. So, and as of today, we are sitting on 200 million AlphaFold models that can be downloaded from their page, like a dragon sitting on its gold. And we are thinking ourselves, well, now we have 200 million structures, and what now? The problem is, it hasn't changed that much. So we are actually in the same stage where we've been 2 or 3 years ago. The good thing is that it's pretty convenient for people who are not so computationally savvy. So if you would have a bench chemist and you would ask him, hey, could you please do a simple docking on a structure that has not been published yet? In ultimate theory, he can now access a pretty good model and then use it for docking to get ideas what to synthesize next. But there's a huge but. We need still a lot of additional refinements to work on that. So if you work with very flexible structures like GPCRs, it's not that you can just take an AlphaFold model and dock into the structure. So basically, the GPCR predictions of the AlphaFold GPCR prediction actually represent inactive structures. So they are pretty close. The binding site is not completely accessible. And if you would dock into the structure, it's just not the right thing, because you can do that, but it doesn't reflect the whole induced fit flexibility mechanism of GPCRs. For solvent-exposed structures, AlphaFold models are extremely good and very reliable. For highly flexible structures, you still need a little bit of refinement here and there. So what you need to do, you need to refine highly flexible structures. Many people do that with MD simulations where they run, I don't know, 50 to 100 nanoseconds and then capture stable conformations of the GPCRs and then use them for virtual screenings, docking, whatsoever. Other use cases, so I had actually a very interesting discussion a few years ago where a company approached us and they wanted to use AlphaFold to actually create libraries. So they wanted to take the AlphaFold structures and then just dock into them. Dock, I don't know, the set was close to 1 million compounds, kind of like a diversity set of what they had on stock, and then just say, well, this is GPCR A, B, C, D. Let's dock those compounds and have then a listing of compounds that are likely active or likely binding there. The idea was if they would do then a cell-based assay and they would find actives like molecule D, then you could go with molecule D into the listings of the GPCR structures and see, okay, this molecule killed cancer cell lines, but did not kill the normal cells. So this may be an interesting target. But what's the target? Then they go to the list and they check, oh, okay, it's in the top 100 ranking compounds for target A, X, and Y. Let's take a closer look at them. So just like to get guidelines to where to look for. But by the end of the day, they realized that docking 1 million compounds at approximately 1,000 structures was not the best thing to do. And then, unfortunately, they had dropped the idea. but maybe next year. So AlphaFold structures are a blessing, but the problems still do remain. As we are aware, structural knowledge about a target is not the limiting step. We had homology model back then. We still do have homology modeling today. And the models are still completely fine that we created with homology models. So basically, AlphaFold is nice, but it doesn't accelerate anything, to be honest. Still, we need to rely on assay data, and this is kind of like the time limiting step where we need to synthesize compounds, we need to test them and do the iterations. So access to better quality structures doesn't solve that much. Again, we don't have enough data. So for a lot of things like ion channels, transporters, GPCRs, there is not enough 3D data, X-ray data available on of PDBs to actually construct good models. So in many cases, homology models are still outperforming AlphaFold. Furthermore, disordered protein regions are still not completely addressed, or absolutely not by AlphaFold. I think the new iteration is supposed to solve that a little bit, but we will need to see how this goes. And still, we are only working with predictions, and this is not a real protein structure. So AlphaFold, nice, but no quantum leap in drug discovery. Okay, now we are entering the field of small molecule drug discovery, and this is where the fun starts. So I asked you, we have invested 16 million of US dollars into drug discovery till 2021. Where are our FDA-approved drugs that come from machine learning approaches? So this was actually the first one that entered clinical trials, and long-acting serotonin receptor agonist, reported actually by collaboration between Excentia and Sumitomo Nippon Pharma. Unfortunately, they never disclosed the 2D structure, not until today, at least, according to my knowledge. But in the patent about this structure, they also showed other stuff that they wanted to patent or claimed by the patent, which we can see on the left side. So a few of you may be aware of haloperidol on the right side, an approved drug, that received approval in 1967. And if you now compare haloperidol to the left molecules, you will ask yourself, well, there is a lot of common features, right? We have this phenyl ring system, the aromatic ring system, and then this piperidine here, and also 6-membered rings here. So yeah, it's kind of like, it's not so surprising that those compounds were active. What's even less surprising is that the shape responsible for this agonism at a lot of receptors actually was reported back in 1913, and about close to 150,000 molecules have been reported, and for this, in arranging into this scaffold. So in the patents, they actually synthesized 350 compounds to come up with TSP1181, and 58% of those molecules share the same shape as haloperidol. And all of those kind of things were actually suggested by machine learning approaches. What's even more surprising is that the other 21 compounds of this 350, 21%, sorry, of the 250 also shares shape and topology with other FDA-approved drugs. So it entered actually clinical trials in January 22. And what was really surprising is how fast this whole procedure was. So it took them only 12 months to come up with this drug candidate, whereas the average drug development procedure takes 4 to 6 years. So this is something. This is quite good. Unfortunately, though, it did not face phase 1, which was reported in June 22. And what I really, really loved, just because it's my humor, is actually the sassy comment by the FDA, because this drug contained insufficient novelty. And if you think about that, if you feed the system with drugs that you are aware that act at this receptor and share this common profile, and this is what you actually patent and this is what you designed, I mean, like, if you feed this kind of stuff into the machine learning model, what do you expect to come out, right? So I'm not so surprised, but this is something that you can kind of like take home as a message from me, at least. So the same collaboration between Excentia and Nippon Pharma also led to the discovery of a dual agonist targeting the serotonin 1A and serotonin 2A receptor. Structure is still unknown, so we cannot discuss the structure here. But if we now compare the 2D structures, so these are the molecules claimed by the patent, and those are approved, FDA-approved drugs on the right side. Again, I'm not very surprised, right? So novelty seems like to be a little bit of a problem for drug discovery. But yeah, it's still undergoing investigations and clinical trials. So maybe hopefully it will receive approval so we can see the 2D structure to talk about that in a few years. So a lot of things are actually in the pipeline. This is just a brief summary. A lot of those companies are actually completely dedicated to applying machine learning and AI approaches to drug discovery. And what we can see here, which is a pretty good sign, is also that a lot of those things actually are able to enter phase 2 trials. And if we check in this green area here, we will also see that even this compound by Recursion Pharmaceuticals also entered phase 3 trials and has been granted the fast track and orphan drug designation by the FDA. So a lot of stuff happening, and there will be a bright future ahead of us. What is good about AI and machine learning, though, is actually that it's able to fill the pipeline of drug discovery. So we can see here on the left side are actually the pipeline that is fed by machine learning and AI. And we can see that now over the years, more and more compounds are entering the pipeline and discovery phase in preclinical trials of pharmaceutical companies. It has reached a level where 50% of pipeline compounds are actually stemming from, 33% actually stemming from AI companies, which is 50% of what big pharma has to offer nowadays. So it is huge, and we will definitely see approvals in close future. I would also like to talk a little bit about the target composition. You can actually see that AI companies are, they're working on a lot of well-established drug targets. As I mentioned, kinases and GPCRs. And there's a good reasoning for that, because for kinases and GPCRs, you have quite a lot of data available that you can use for your machine learning. So everything is more or less even published and available on a lot of websites. So this data is something that you can use, but since everybody has been working on that, so, it's no surprise. Still, they play it safe here. So AI companies also try to target something that is a valid target, because this is where they are more likely to come up with a success story to obviously keep on going and continue their agenda. So this is a safe bet, but nevertheless, there are also some first-in-class targets that you can see here, like a DNA helicase. So there's a lot of stuff going on also on novel ideas. Let's call it like this. Big Pharma also works on well-known targets, but as you can see here, also difficult receptors like transporters and nuclear hormone receptors are also addressed by the top 20 pharma companies. Still, what's really surprising is that AI programs complete the whole the early stages of the drug discovery process a little bit faster, saving close to 1 year compared to the pharmaceutical companies. So 4 years versus 6 to 5 years. The problem we saw in the previous slides, that a lot of structures are very similar to what is already known. And what you should know about here, this is kind of like a representation of a chemical space. This is the uniform manifold approximation and projection. So what you're trying to do here, you try to flatten the whole multiverse of the chemical space, a lot of parameters, into a 2D representation. And what you can see here for this target, here's the kinase, that distribution of non-AI derived compounds compared to AI derived compounds. And what you can see here is that AI derived compounds are actually always in the middle of what is already known. So they kind of like not extrapolating into new areas of the chemical space, but they stick to what is already there, what is already published, what is already aware of, which is no surprise because, again, it's reliant on the input data. It should be also a good thing to ask, well, are medicinal chemists better than AI in regards to being more creative and to produce novelty? There's a beautiful review on that, where researchers at Novartis were challenged with drug discovery tasks, and everything was documented. It's a really nice read. So please do it afterwards or in the new year. And what was actually addressed there, that even chemists express a bias. So you always, if you talk to a medicinal chemist and you ask him, how would you improve this compound? He comes up with the sulfonamide. So it's no surprise, right? But experienced chemists do have an opinion about stuff, and they push this opinion. So you are human, and since you have your own experiences and your knowledge, you try to push particular ideas, and this is also where you limit yourself to targeting a particular area of the chemical space. So the deduction from this whole publication was actually that at least 2 or 3 chemists with different backgrounds should actually work together if they would like to identify novel and interesting series of compounds. So since time is running up, I'm accelerating a little bit and dive into the antibiotics discovery field. So since 1918, no clinical antibiotics have been discovered by high throughput screening. And this beautiful publication here actually used deep neural networks to predict novel compounds. So what they did, they used a neural network to actually capture the whole superstructure of a molecule to identify molecular features that are responsible for antibiotic activity. And what they did, they purchased 2,300 compounds and tested their activity against E. coli. And they simply said, yeah, it's active or inactive. So the molecular representation covered the whole chemical structure here, which was then used to predict novel compounds. And what they did then, they screened another library consisting of 6,000 molecules from the drug repurposing hub, and they let the model score those. And what was really surprising, 51 of the top 199 predicted hits actually were able to inhibit the growth of E. coli. And what's even more surprising, that the score that the model provided to each compound also correlated with the inhibitory activity. So given the fact that they had a lot of compounds, to put 3,000 to train the system, and then use this one as a test set, made this whole model pretty powerful. A little bit of a downside though is actually, from my perspective, the novelty here. So this was the most active compound with an MIC of 2 micrograms per milliliter. And it was, the compound is called halicine, which is actually an C-junction N-terminal kinase inhibitor. And if you may recall the structure of metronidazole, it looks a little bit similar to that. So this Tanimoto similarity is actually referring to metronidazole. What surprised me, on the other hand, is though the another set of compounds. So they also screened the zinc database, consisting of 107 million compounds back then, because they really wanted to have something novel in their hands and not only repurposed drugs. And then they purchased again 23 compounds, and 8 were active, those 8 here. And what I really found interesting is actually this particular compound. Zinc 0001000327716, this guy here. And if you compare it to the closest neighbor, enrofloxacin, with a tiny mode similarity of 0.39, you will obviously see a lot of common substructures. So, I mean, like, this is a fluoroquinolone, so this is like the most typical gyrase inhibitor profile you can imagine. What's surprising though is the substitution of the fluorine group here for a nitro group. I'm actually surprised that it's tolerated, and it was predicted to be also pretty stable metabolically wise. But what is the most surprising thing for me is actually this part here. So if you take a look at that, you would recognize a sulfonamide partial substructure. So something that was actually discovered back in World War II, or at least in the 40s, by Nazi Germany. So an active class of antibiotics, which were not pursued afterwards for humans, but are still in use for animals. And this is actually something that interacts with folate synthesis in bacteria. So this is actually a dual mode of inhibition. It may have a dual form of inhibition. And this is not something that you would design as a medicinal chemist. So you would come up maybe with this, but you would never, ever, ever come up with the idea to actually couple this together through this azo-like substructure to the sulfonamide. And you can actually see how potent this compound is. So from my perspective, very interesting, because this is not something that a human would come up with, but active. Another story is actually the targeting of bacterial RNA. I think I will rush through this. So basically, they did a lot of things to actually predict actives. They started with 791 molecules containing an phenyltiazole, and then applied different machine learning methods, including decision trees and convolutional neural networks, to actually predict new active compounds. Actually, I'm missing here. So they predicted compounds, and then finally they synthesized 10 novel compounds, and 3 out of the 4 predicted good binders also inhibited the target. So you can actually see whatever they did here based on the SMICE representation, the decision trees, and so on and so forth. So there was a lot of effort involved in that. And they were able to predict 3 good compounds. 75% is an amazing hit rate. I cannot downgrade this. But what you should pay attention to actually the docking values. So now in bold, the active compounds here. And if you would now check the docking values, they actually, obviously, the first candidates you would actually pick based on docking values. So as I don't want to kind of like downsize the efforts and the outcomes of the machine learning approaches here, I would actually argue if you would need to do everything involved here to come up to something that is, yeah, basically the same result that you could achieve with docking. And even if you would use merely docking and even supplement with a second docking algorithm to kind of like do an orthogonal validation of your results, I mean, you would end up with the same results. So from my perspective, it's good that we investigate those kind of things, but we are not there yet where we are better than simple docking procedures. So my take-home message for this part is actually machine learning is still a closed ecosystem. So you can only mine from the knowledge it was provided with. So don't expect apples if you seed tomatoes. So you end up with the same thing that you've put into the system. Yet the classification in actives, like actives versus inactives, says the cost of the results. So if you use only one scaffold and one molecular compound series to define what's active, you will never reach into novel chemical space areas. You will always end up with very similar compounds. So my suggestion, my recommendation is actually, if you want novelty, you have to go wild. So supplement your model with non-AI content. So a lot of our customers actually mine the chemical space, the accessible chemical space, with our tools to check what is available. They have a proposal for machine learning, a structure, and then they dive into the chemical space using different algorithms to check what's the closest compound, what's the closest neighbor to what the machine learning approach would like to do. because this is also, once you search within our commercial chemical spaces, you end up with results that are commercially available, tangible, and can be also bought for a very reasonable price. So those are accessible results, whereas for whatever the machine learning comes up with, you would still need to check if this is synthesizable or not. Another idea factory, as I would call it, is actually using 3D methods. If you have a target there, use it. So use it for docking, use it for generating ideas of what you like. It doesn't have to be active, but where you say, well, this particular structure is of interest because it interacts beautifully with my binding site. Also, CISA allows you to, and our other tools like height allow you to also attribute different parameters like individual contributions of atoms from the docking procedure to also supplement your procedures with additional data. So FastGrow, also beautiful, met canvases of CISA to generate really novel ideas to complement the binding site and to increase the binding affinity. So if you have the luxury of working at a 3D structure, use it also to kind of like just generate ideas and then feed them into your machine learning approach. Yeah, drug repurposing is also pretty interesting thing. So baricitinib is an JAK1 and 2 inhibitor that was designed and developed by Eli Lilly, which was actually predicted by benevolent AI to be used in COVID-19 infections. And they used a deep learning approach that took them only 48 hours to come up with baricitinib as a potential drug to treat complicated COVID-19 infections. And for this, they use actually multidisciplinary data, so also patient data and other things. So drug repurposing with AI is something that will be also pretty prominent in the upcoming years. So last but not least, this is actually the last setting, is actually how is AI penetrating the modern industry. What I really like is this collaboration, Melody. It was founded in 2019 and ended in July 22, and was the world's largest collaboration of pharmaceutical companies to actually improve their models to make more accurate and efficient models. It was actually co-funded by the European Union with merely 18 million US dollars, not euros, sorry, it was euros. And those 10 companies were part of the whole setting, and they also had technology partners listed down below. And this is what they achieved with 18 million euros. So the models improved by 4% in regards to the pharmacological and toxicological profile, as well as the prediction towards actives and non-actives. Multi-partner models showed a 10% increase in its application domain, and collaborative models were typically 2% better at estimating values of activities. So it's up for you to decide if this was a huge success or not. Nevertheless, it pushes the idea of actually sharing data between companies, which is required to improve the models in the future, and also to kind of like contribute to the whole idea of actually making the research area transparent. I have to admit that I'm a huge fan of the Merck group, because they are the ones that, almost religiously apply machine learning in a daily environment. And I really like this phrasing of Friedrich Schrippmann, predictions must be easy to access. So what chemists actually do at Merck, they have this beautiful web interface where they can draw molecules, design them for their projects, and they get instant feedback of what the different models of ADME properties think about this structure. And this is where you can also see this traffic light scheme that it tells you, well, you should work on that because this is unstable, it doesn't make sense to actually synthesize this compound. And they do it for a lot of things, a lot of ADME properties and also physical chemical properties. And the outcome is that predicted bad compounds will not be made. So you save money, you save time, you save even like discussions. Should we do this? Should we do this? Shouldn't we do this? It saves a lot of time. And it's kind of like, because everybody has access to this kind of thing, it improves the whole quality, which is also reflected by the numbers. The log P measurements have dropped the required retestings of data. So you save time for assessing the compounds for their quality. And also retesting is also triggered automatically. So if something doesn't align too well with your predictions, you try to check if somebody made a mistake to actually still use this data to feed back into the system to make reliable in silico predictions. And this is an iteration process. So I think he mentioned that they update the models bi-weekly or weekly, depending on the model, to actually reflect the newest insights from their data. So they do it very religiously, and is quite implemented into their workflows. Okay, we've reached a beautiful stage where this will be the last slide of the presentation. So the take-home messages for you: AI is there, but do not expect miracles. It's still in the training shoes, and we still need to learn. And there was a beautiful phrasing during the pandemic in Germany, where the German health minister said, there will be a lot of things that we need to forgive each other. And I think this also applies for the machine learning and AI field. We are still learning, and it's a work in progress. So mistakes can happen and will happen, but we should be open to learn from them. We have to accept the fact that the large-scale process is very complex, and collecting and processing enough data to actually supplement it with enough information is close to impossible. Also, given the fact that we don't even know what biological parameters are relevant for us. Nevertheless, AI is a valuable tool. We can do a lot of things like admin predictions with that. It's like the most viable tool in this regard, I would assume. And yet we should still focus that AI should not be considered at, like, it should more from the what can be done perspective and be fixed towards the what should be done with AI to arrive at more efficient operation with that. So before we can actually save money, we should handle biological data better. This includes handling data better between different departments and also the open access data sources. And yeah, it's a process to learn. I mentioned that already. But the huge thing is actually that the estimates suggest that AI could reduce the time to market new drugs by 50%. So there's still a lot of things going on, but we need to wait, and it's up to you to decide how you perceive AI from now on. So with this, I actually end my presentation, and thank you for listening, and I'm happy to answer the questions if I am able to do so. Thank you for listening so far.